Thanks a lot for the organizers for giving me this opportunity to present my work, and thank you all for joining. Today, I want to present an approach we developed for multimodal analysis of cell-free DNA whole genome sequencing data from pediatric cancers with low mutational burden. My name is Peneder. I'm a bioinformatics PhD student in the Tumor lab for epigenome-based precision medicine at the St. Anna Children's Cancer Research Institute in beautiful Vienna, Austria. I want to start by giving you a brief introduction into liquid biopsies. In patients with cancer, the tumor releases cell-free DNA fragments into the bloodstream via secretion or during apoptosis or necrosis. If we then collect a blood sample from a patient with cancer, a fraction of the cell-free DNA that is contained in this blood sample will stem from the tumor. Now, of course, the key challenge for using liquid biopsies in oncology is then to distinguish which fragments stem from the tumor and which stem from healthy cells. And usually this distinction is done via the detection of somatic mutations in adult cancers, but we are working with pediatric cancers, so we're taking a slightly different approach, as you'll see in a second. But first, I want to give a motivation of why we really care about bringing liquid biopsies closer to clinical practice. Solid pediatric tumors are a challenge for clinical monitoring. They require invasive biopsies or imaging. They cannot be performed every time the patient presents at the clinic, so clinicians are facing limited information. If they had access to high-resolution data, such as liquid biopsies can provide, this could lead to a better treatment outcome. For example, by allowing early detection of relapse than is possible with traditional methods, as you can see here below. Liquid biopsies only require blood draws from patients, which are minimally invasive, and therefore can be performed quite frequently, which is a great advantage. It's important to know that pediatric cancers are a bit different from adult cancers. They have a lower mutational burden, but many pediatric cancers have distinct epigenetic properties. And this is true also for Ewing sarcoma, which is a pediatric solid tumor that affects bone and soft tissue that we're using as a model disease in this study. Ewing sarcoma has few recurrent mutations, and it's a so-called enhancer disease, where a fusion oncogene drives the disease and induces a disease-specific epigenetic signature. The aims for our study were to interrogate the epigenome of Ewing sarcoma and other pediatric tumors via liquid biopsies, and to develop mutation-independent epigenetic biomarkers to detect and classify tumors. To this end, we collected a cohort of patients with Ewing sarcoma, patients with other sarcomas, and healthy controls. We then analyzed this data using a fragmentomic approach. And this is based on the fact that while cell-free DNA is released, it gets fragmented. And this fragmentation doesn't occur randomly, but preferentially at stretches of DNA that are not protected by nucleosomes, as you can see here. Therefore, by knowing the location of cell-free DNA fragments, we can infer where nucleosomes were positioned, and therefore can conclude the epigenetic states of the cells that give rise to the cell-free DNA. Therefore, we collected liquid biopsy samples, extracted cell-free DNA, and sent this cell-free DNA for paired and whole genome sequencing. We then analyzed this whole genome sequencing data on various different levels. First, we looked at the global fragment size distribution. And there we found, similar to what has recently been reported for adult cancers, that cell-free DNA fragments that come from pediatric tumor cells tend to be shorter than those that come from healthy cells. And this makes the global fragment size distribution a useful pan-cancer marker, because cancer patients usually show distributions that are shifted towards shorter fragments compared to distributions from healthy controls. Next, we looked at the regional fragment size distribution along the genome. And there we found that cell-free DNA fragments that come from regions with open chromatin tend to be shorter than those that come from regions with closed chromatin, which is another useful marker. And finally, and this is going to be the focus of my talk today, we looked at the fragment coverage at regions of interest. So what we found is that at regions of interest, or regulatory regions, such as DNase hypersensitivity sites, there are fewer nucleosomes protecting the DNA from fragmentation if the source cell of the cell-free DNA is epigenetically active at these regions. And therefore, because there is less protection, we will see a decrease in mapping fragments at the center of these active regions, which we can then observe as a drop in coverage around the center. And to quantitate this phenomenon, we developed LIQUORICE, which is the tool to detect tissue and cancer-specific epigenetic signatures in whole genome sequencing data from liquid biopsies. LIQUORICE takes as input two things: tissue, a set of tissue or cancer-specific regulatory regions, for example, DNase hypersensitivity sites or enhancer peaks, and the mapped reads of your sample of interest. LIQUORICE then calculates a bias-corrected composite coverage signature of your sample and extracts quantitative metrics such as the area or depth of this coverage drop. LIQUORICE works as follows. It starts by splitting every region into bins and then calculates the coverage and the number of bias factors for every bin. These bias factors, for example, GC content, are important because they influence the coverage without being related to the epigenetic properties that we actually care about. So in the second step, we use machine learning to correct these associations between bias factors and coverage. Third, we average the coverage signal over all regions in our region set in order to obtain a more robust signal. Finally, we quantify this coverage signal by fitting 3 Gaussian functions and an intercept to this coverage drop, which allows us to extract metrics such as the area over the curve or the total depth of the dip, which you can then use to make comparisons between samples. And when we do this for our own cohort, starting with Ewing sarcoma-specific DNase hypersensitivity sites, and this is a set of regions that we obtained from prior literature, we found the following. First, looking at the healthy controls and the non-Ewing sarcoma samples, we find that they have a rather flat profile with even a small peak around the center of these DNase hypersensitivity sites. On the other hand, our Ewing sarcoma samples, and especially those for which we have independent genetic evidence for tumor-derived DNA, we see that they show a drop in coverage around the center of these regions. And as you can see, this is a quite specific signal. So only Ewing sarcoma samples show this drop in coverage. None of the controls show this drop. As another control, we performed the same analysis for a set of universally accessible DNase hypersensitivity sites. So these are sites that are accessible across all different kinds of tissues. And as expected, we don't really see any difference between our groups of samples. It turns out that the Ewing sarcoma specific DNase hypersensitivity signature is a very quantitative marker for tumor content, as you can see on this plot on the right. So on the y-axis, we have the LIQUORICE inferred coverage signal, and on the x-axis, we have the percentage of tumor-derived cell-free DNA as inferred from independent genetic markers. You can see that for our Ewing sarcoma samples, this correlation is very high, reaching a correlation coefficient of 0.89. Whereas for non-Ewing sarcoma samples, this correlation is virtually non-existent. I would like to mention that LIQUORICE is applicable also to healthy tissues, such as blood or liver, and we have successfully applied it also to a small number of samples from other tumors, such as rhabdomyosarcoma samples, which points out that LIQUORICE is really more broadly useful than only applying it to Ewing sarcoma. Next, we took a number of fragment-based metrics that I have introduced earlier, the global fragment size distribution, the regional fragment size distribution along the genome, our LIQUORICE inferred coverage drop, and read depth as a marker for copy number aberration that we get kind of for free on top when we do whole genome sequencing of cell-free DNA. And we took these metrics and fed them into machine learning classifiers as features and trained these classifiers to distinguish Ewing sarcoma samples from healthy controls. And as you can appreciate in these receiver operator curves, the metrics work quite well as features for distinguishing these two groups of samples, with the best features being the regional fragment size distribution and the read depth, reaching 0.97 as a ROC AUC value. So this is quite nice because this shows that tumor detection works quite well with these fragment-based approaches. Second, we trained our machine learning classifiers to distinguish between Ewing sarcoma samples and samples from patients with other sarcomas. And as you can appreciate, especially our LIQUORICE-based coverage signature performs very well here, reaching ROC AUC values of 0.92 and clearly outperforming the other metrics. As the final result that I would like to show today, We also found that the tumor detection based on the Ewing sarcoma-specific epigenetic signature, as inferred by LIQUORICE, is associated with poor survival. What we did here is we took our samples that we have from patients with Ewing sarcoma at diagnosis and with localized disease, and split these samples into two groups. Those for which we did not detect tumor-derived DNA based on LIQUORICE because the tumor content was very low, and those for which we did detect this signature. And as you can appreciate in this Kaplan-Meier plot, the group for which we did detect tumor-derived DNA performed much more poorly in terms of relapse-free survival and also overall survival compared to the other group. This makes the LIQUORICE inferred Ewing sarcoma-specific epigenetic signature a quite interesting prognostic biomarker that we will analyze in future studies. I want to use this opportunity to advertise our tool, LIQUORICE, a little bit more. So as you've seen, LIQUORICE is a powerful tool to detect tissue and cancer-specific epigenetic signatures in whole genome sequencing data from liquid biopsies. And it will be available very soon as a command line tool and Python package via Bioconda. At the moment, it's available for manual installation on our GitHub account. This is github.com/peneder/LIQUORICE. And you can also write me an email or get in touch at the poster session if you're interested in using LIQUORICE on your own samples or if you have any further questions. If you want to use LIQUORICE on your own data, what you would need to do is collect cell-free DNA samples from a number of patients and controls. And then for every sample, run LIQUORICE with the mapped reads of these samples. And as an input, you also need to give it a list of region sets that you're interested in. This would be, for example, Ewing sarcoma-specific sites or blood-specific sites, things like that. And you can then run this convenient summary command that provides you with an overview of the epigenetic signature in each region set for your healthy controls and for your patient data. And it also provides you with a number of plots for the bias correction and additional information. Potential application areas of LIQUORICE include cancer detection, classification, monitoring, and survival prognosis, as you've seen before. It could also be interesting to apply it to identify cancers of unknown primary or to locate metastasis. And it could also be useful for tissue damage monitoring, for example, after transplantations, heart attacks, or to detect therapy-induced toxicity or to study tissue damage due to COVID-19, for example. Yeah, this brings me to my summary. So I hope I could convince you that fragmentomic analysis of cell-free DNA whole genome sequencing data is kind of a one-size-fits-all assay that allows us to do various things. First, it allows us to detect and quantify tumor-derived DNA independent from somatic mutations. It allows us also to distinguish between different cancer types based on their epigenetic signatures. It allows us to predict disease course, and it could be useful for disease monitoring and treatment response assessment. Importantly, whole genome sequencing of cell-free DNA is very feasible in a clinical setting. It requires less than 10 nanogram of cell-free DNA as an input. It profits from rapidly falling costs of whole genome sequencing, and it does not require access to primary tumor tissue, which is also a great advantage compared to traditional methods. If you're interested in our study and would like to read up on the results that I presented today and many more, you can check out our publication titled Multimodal Analysis of Cell-Free DNA Whole Genome Sequencing for Cancers with Low Mutational Burden that was recently published in Nature Communications. Yeah, finally, I want to point out that we are currently looking for a bioinformatics master or PhD student to join us to work on liquid biopsy data analysis or other exciting computational challenges in pediatric precision oncology. So if you're interested, visit tomasolab.org and get in touch. I want to thank mainly Eleni Tomaso and Christoph Bock, who are my supervisors for this project, and Adrian Stütz, my colleague, who did a lot of genetics-based work and experimental work for this project. And I also would like to thank a lot of other people, both in our institute and our collaboration partners, who provided us with samples and expertise. And finally, I want to thank you for your attention, and I'm happy to take any questions.